about you, so don't worry. És un plaer de comptar avui amb la presència entre nosaltres de la doctora Deborah Power. La doctora Power va ser escollida electa com a membre corresponent l'any 2022 aquí a la Reial Acadèmica Ciências i Arts de Barcelona i fins avui no hem tingut l'oportunitat de tenir-la entre nosaltres i és un plaer que avui hagi pogut venir. La doctora Power és professora i investigadora del Centre de Ciències del Mar a la Universitat de l'Algarve, a Portugal, i centra la seva recerca en els camps de l'endocrinologia comparada i la biologia molecular. Endocrinologia comparada des d'una aproximació molecular, per entendre'ns. De fet, ha desenvolupat una intensa activitat investigadora integrant recerca de peixos i de mol·luscs, com després veurem. I sobretot el seu programa de recerca s'ha centrat en l'estudi de la regulació hormonal endocrina de l'homeòstasi i això enfocat a diversos nivells d'organització. Que això és una cosa que no és massa habitual perquè la tendència actual és especialitzar-se en una cosa molt concreta. En canvi, la doctora Power, els seus estudis sempre ha intentat abarcar des de l'organisme en conjunt, punt de vista organísmic, diguem-ne clàssic, fins a l'escala molecular, passant per totes les escales. Ha estat presidenta de la Societat Europea d'Endocrinologia Comparada, ara ja no ho és, i entre altres mèrits, que en té molts, però l'any 2012 fou distingida amb el doctorat honoris causa per la Universitat de Göteborg, a Suècia, per les seves contribucions a la biotecnologia i a l'aqüicultura. Ha estat reconeguda com a professora distingida per diverses universitats i institucions internacionals, com la Universitat de Shanghai i l'Institut de Tecnologia d'Irlanda. Bé, com deia, avui tenim l'oportunitat de rebre-la personalment a la nostra acadèmia, la qual cosa és un goig, i de gaudir de la conferència que ens ha preparat i que porta per títol From Molecule to Ecosystem and Beyond the Anthropocene. Doctora Power, tiene usted la palabra.
So I'll take the time. So thank you very, very much for the presentation, and I thank very much the invitation to be present, to present some of this work, but also for the election, which I feel very honored to have been elected to join this esteemed academy. So my title, I guess, it's deliberate because I'm actually going to talk relatively little about the Anthropocene. But if I actually say to people I'm going to talk about mollusks, probably no one will turn up, because for most people these are rather uninteresting things unless they're on your plate for eating. So I will try today to shine some light on the importance of these organisms and maybe on what they can tell us and how much is still left to be done. So I will pass through some of this, but I've been a bit liberal with my title. So before I go into my talk, just to give a very brief bio of who I am or what I've done. So I actually, as you probably realized, I'm from the UK. I actually worked initially, I did my first degree, an honours degree in London. This was nutrition with physiology. Then I went to the Faculty of Medicine and did my PhD in Liverpool. And then from Liverpool, I was recruited as the first female higher scientific officer in the Ministry of Agriculture, Fisheries and Food. And then, of course, I met my husband, and he was from Portugal. So either I stay in the UK or I go to Portugal. And at this time, which is effectively 38 years ago, the science and education scene in Portugal was much less developed. There were a lot of opportunities in the sense that there was very little done. And so I decided that I would take a chance and I would actually go to Portugal and carry on the development of my career there and contribute, hopefully, to the development of science. And so this is why I have these two maps, the UK and here Portugal with Spain, of course. And now I am the professor of biotechnology and I'm leading this group in comparative endocrinology and integrative biology. So this was a challenge because we had to set up from zero, absolutely everything. There was nothing at all. As Xavier pointed out, I am a comparative endocrinologist. So actually, when I started putting this together, I started to realize I've actually been working on an awful lot of different kinds of organisms: pigs, sheep, mice, fish, insects, mollusks. So the whole concept of the comparative model actually is very useful, and as I will point out later on, has been really a driving force within certain areas of science because of the power that comes from making comparisons. So I will touch on that today. So the talk in itself, what I wanted to start by doing is just giving an overview of the mollusca, the diversity and importance. I know we have evolutionary biologists, so this will be nothing new for them. I want to point out and explain the success of the bivalves, and this is because of this very important evolutionary innovation, which is the shell. But of course, this shell then becomes a point of stress within a changing ocean. So there is a lack of information about the processes behind the regulation of the production of the shell. And when I started to look at this, I realized we can't really understand how they're going to respond to changing environment if we don't actually understand the physiology and the regulatory processes of the organism. And so therefore, this is where I started to look in more detail and then subsequently try to put this in the context of the changing ocean, where we see an increase in acidification. So to start with the diversity, so the mollusks are an enormously diverse group of organisms. This phyla is the second most abundant of extant organisms after the arthropods. And as you can see here, I mean, most of us are familiar with a number of these different organisms, things like the bivalve, the gastropod, the octopus, the sepia. So they're very, very diverse and very, very numerous. And therefore, they have managed with their genome during evolution to acquire very diverse forms, occupy very diverse niche, and to be very, very successful because you first see them emerging in the Cambrian period. They've been known and have been an important source of a range of different things for man. So, for example, the Phoenicians extracted dyes for cloths and for painting and for artwork. Jewelry made from shells was very, very important. Also, it was used as a currency. And then, of course, it is a source of food. It's a very popular source of food for many people. But on the negative side, we also know that they can be vectors for disease, for pathogens. So, for example, liver fluke, where the gastropod serves as part of the life cycle. The poisonous cone snail that can kill you, but nowadays, through biotechnology, it's actually being used for anesthetic or for pain relief, given in the correct doses. And then, of course, the mythical kraken. So it's something that's always fascinated the human population is in fact this really diverse set of organisms. But when we come to science, here we also see that they have been of fundamental importance for scientific insight. A whole series of the mollusks that have contributed to fundamental understanding that is now, of course, applied in biomedicine, but started off in these very simple-looking organisms. So considering, for example, the gastropod, these Eplisia and Limnia, these were of key importance and remain very important in neurobiology and are very important models for learning and memory. If we consider the squids, the understanding of the action potential, nervous transmission, This was all done in squids because of these very large axons, and Hodgkin and Huxley, of course, gained the Nobel with that. The brachiopods, we can find fossils. Stephen Jay Gould in the Burgess Shale looked at this for aging, some of the fossils there. And so the brachiopods in terms of geology have been very, very fundamentally important for understanding the time scale when looking at different sediments. But what I want to talk about are the bivalves, because this is the group that I have been specifically working on in relation to the shell, which are primarily considered as important ecological models, and also for ecotoxicology, they are considered to be very useful in that context. So the bivalves, why bivalves? Well, bivalves because they have these two valves, so two shells, and this shell is produced by the mantle. So this is of course, this clam here, you can see this very ornate, very large, very developed mantle, and a lot of sensory tentacles here. But all of the bivalves have the mantle. Not all of them will produce a shell, but most of them will secrete a shell, or these two shells, and this will serve as protection. And this is the innovation that is considered to explain in part why they have persisted so long. The shell, even though we look at the diversity of bivalves, what we can see is the shell is very, very conserved. So it's primarily calcium carbonate. So unlike the vertebrates that use hydroxyapatite, so phosphate and calcium, the bivalves and other organisms within the mollusks are using this calcium carbonate. And this is where the first point linked to the ocean acidification comes in, which is related to dissolution and the solubilization of the actual shell, or the inability of the larvae to form the shell. So this is one of the key points in terms of understanding potential impacts. If we consider them from a more global perspective, the bivalves gain this even wider and more important function in the ecosystem, because they're actually filtering massive amounts of water. And because they're filtering so much water to obviously acquire the food, they're also improving the water quality. They tend to form these very large reefs, so they tend to aggregate together. And this creates ecosystems for other organisms in the environment, which can be marine or fresh. And they're considered environmental engineers because they actually create these artificial barriers and reefs which contribute to control erosion. Of course, they're a food source for a whole series of different organisms, and also for humans. More recently, they've been looked at in the context of this carbon sequestration as potential mechanism by which you can try and control this imbalance that we have currently in terms of CO2 production and absorption. And just to highlight, there is this very extreme diversity, because this, when we come back to talk a bit more about the shell, is a very important point, is to understand how you can create so much diversity and yet retain some very common characteristics. The main organisms I've been working on are these two here. That's the oyster. So this is a bivalve, but it's one of the very few asymmetric bivalves, because the two shells or valves are not the same size. And the mussel, which has this very beautiful symmetry. Both belong to the Pteriomorpha, but to different groups there, Australia and Mytilids. So, and then finally, where they now are gaining very much importance in terms of societal is linked, of course, to the fact they're considered to be lower trophic species. We know that the terrestrial production systems are no longer able to supply the food that we require. We have had a devastating impact already on biodiversity loss, pollution, deforestation, soil degradation, coming through this practice of terrestrial agriculture aiming to feed the planet. And the rate of growth now of the population is such that in a very little time, it will be no longer sustainable to produce food from terrestrial sources. So the marine domain now is the next big frontier. This is where the food is supposed to be coming from. So it makes this a very important time, because we hope that when this exploitation occurs, as is foreseen in large scale, that there will be protection in place but understanding the organisms, understanding how they are within the environment and how they respond to changes in the environment is a very, very important aspect. And so this is very much how I contextualize my work, although the fundamental scientific questions may be what excite me. When I'm trying to sell it, of course, politically, you need to have a very clear context from a socioeconomic perspective. And this, I think, is fairly compelling. So the shell. So, you know, really what is the shell? And how did this innovation contribute to this success that we actually see? And knowing about the shell, what can that tell us about the likely impact as we proceed and the climate continues to change, be it through human or any other mechanism? The reality is there is a change, and that change is impacting organisms. So this is where, this was, I guess, our starting point, was really this question linked to how do you produce a shell? How do you maintain a shell? What are the factors behind that? And what are the potential tipping points? So up to what point can the animal sustain shell production within a changing environment? And of course, as is always the case, we always very much depend on our colleagues and younger and older colleagues to collaborate and to produce the data that someone like myself will be presenting. So the shell in itself, if we actually look at the shell, so I have the two examples here on the right. On the left, I have the oyster, on the right, the mussel. And most people, I think, are quite aware of how different these shells are. The oyster is quite chalky, quite soft, and the actual mussel is quite brittle, quite hard. So they're really quite different, but they're both being produced by the mantle. So it's this similar tissue because it looks quite similar in the two of them. And then when we come and look at just some of this diversity, form, shape, function, it's really, really remarkable because it's enormously diverse. And yet, when it comes down to it, it all consists of this organic matrix, less than 5%, so it's a very small part. It creates a framework, and then primarily calcium carbonate, approximately 95%, and that's what's in all these shells. So it doesn't matter what shape, color, size, form, this is what they contain. And so how do you explain the difference between these different shells? So when you go in, zoom in and look at the scanning electron microscopy, what you can see is these main crystalline structures. You have foliate, then you have this foliate sort of random, then cross-laminar, nacreous, prismatic, and then the nacreous. So the different form of these crystals, the different disposition in the shells, the presence or not of more or less magnesium and other trace amounts of other minerals can affect how the shell appears and the characteristics of the shell, the mechanical and so on. So here we are, calcified shells, enormously diverse, consist of crystallized calcium carbonate, and basically that's what you have there summarized on the screen are the different kinds of crystals. So when you go look in the literature, there's material studies because, of course, the military think this could be very interesting, this kind of material, because of the characteristics it has. So there's military research looking at the structure. The people that work on ocean acidification, the ecologists, are looking at it from a dissolution perspective. How quickly does it dissolve? But actually understanding how it's being produced, there's much less information about that. There was this boom more recently linked to ocean acidification to try and define maybe what constitutes a shell, but there's no real consistent data, and therefore this is our starting point in our study. This is the cartoon that we have at the moment, and the cartoon, again, very diverse, but this is the basic model. So this cartoon shows the shell on the top, and this is the mantle on the bottom. It's primarily connective tissue, and then you have this monolayer of mantle cells. So these are the secretory cells. And we have this bioreactor in here, which is the so-called extrapallial space. So in that space, the shell is being produced. Now, the fascinating aspect is things like bivalves and mollusks. They're not very good at regulating their body fluids. They don't regulate temperature, but they also are what we call osmoconformers. So they tend to have a composition which is similar to the environment in which you find. So of course, this immediately raises this question: how can they make a shell if they're like the environment they're found in? And this is where this very special chamber here comes in, the extrapaleo space. And I think that the textbooks are not truly representing the situation with regards to how the bivalves and mollusks function. But that's another story. So basically in this space, the bivalve is able to create that space. It's a unique space, and this kind of process that we see here is ongoing. This would be the seawater. So the CO2 is pumped in, we get the carbonate going in, and then we have a whole series of different channels and pumps here that will be pumping in to the extrapaleo space. This is the shell up here. And effectively, through these combination of these different pumps, things are moving into the extrapaleo space, and then we get the chemical reaction that's generating the calcium carbonate, which then deposits as a crystal. So that's basically the state of the art. And that's a model, and it's not really been very tested, but this is how we think the shell is being produced. Then we superimpose on that what we know in terms of chemistry in relation to what's happening in the ocean, where we're seeing an increase in CO2, we get an increase in carbonic acids, increased acidity, we're getting carbonate and a liberation of protons, so we're getting more acid again, and we have this equilibrium that is starting to shift in this direction, and therefore it's getting more acidic, but also the carbonate saturation is changing. So this is effectively creating an energetic problem for the animal to try and pump, and creating difficulty to acquire and build the shell. So that's kind of the eggshell of where the problem exists in relation to OA. And that's what people are trying to look at. Of course, the problem we have is this diversity, massive diversity. And then when we look at a geological timescale, well, you might just say, you're just talking rubbish. Because actually, if we look here when the Cambrian, and you look at the CO2 levels, and you look at the temperatures, and here we are in current day, there's a lot of fossils there. They already emerged in these very high CO2 situations, so high acidity in the water. The temperatures were very high. There have been fluctuations, but they have persisted. And here we are, current day. It's the second most abundant phyla in the animal kingdom. And there is this massive diversity. We have much lower CO2, much lower temperature. So what's the problem with ocean acidification then? I think the main problem is the speed. While they've had millions of years to evolve and acclimate or adapt to these conditions, nowadays that change is much quicker. And therefore, this question of tipping points and what the animal can resist in terms of its physiology become very, very important. So now I'm going to move from the summary and this first introductory part and try and explain a little bit what we've been trying to do to try and understand a bit better this process of how the shell is actually being built. So to summarize, the calcium and bicarbonate, they're mobilized in the mantle edge and they are used for biomineralization. We have this framework, which is created out of complex polysaccharides like chitin and a set of proteins called shell matrix proteins, which are rather unknown. And then we have this other aspect, which is the need to coordinate, because if you're a bivalve and you have two symmetrical valves, you need to grow them at the same speed. And this is a complete blank in terms of what might be responsible for that growth. And we can say that although we have some shared elements between different bivalves, there's an awful lot of species-specific processes ongoing, and there are an awful lot of different architectures in the bivalves, and therefore the response to change is going to be very different. Okay, so there's a tendency to think marine calcifiers, they're all going to respond in the same way. They're not, because there's quite a big disparity in the shell characteristics, but also in the kinds of proteins and the process of building the shell. So understanding how to build a shell, if the two valves are growing at the same rate, and they're growing maybe 1 millimeter over a month, that's not a very easy thing to look at. So what we did was try and think about ways of being able to go in and identify what might be different between a growing and an ungrowing shell. And so here is where we thought about different kinds of bivalves, those that are symmetrical and those that are asymmetrical, because your hypothesis might be an asymmetrical bivalve, the faster growing valve will be the round one, and the slower growing valve will be the flat one. And therefore, there'll be a growth differentiation between them, and potentially we can identify molecules that might explain that. Now, this is nothing new, because Krog in 1920, that won the Nobel, he had already identified the importance of comparative models. And he also identified the fact, of course, that there may be particular organisms with particular characteristics that will give you a keyhole into the problem. And this is what we try to do here. Of course, what he didn't say was that there are no tools, there are no molecular or no animals available, there's no life cycle. So you choose a model, but then you also, what you gain from the particular functional characteristics that may respond to your question, you lose because often you don't have access to reagents and you have to start from the very beginning. So this is what we did. And I just summarized here an overview of kind of the questions and the problem that we have. So the first question here, so we said, okay, here's a perfect symmetry in a mussel versus asymmetry in an oyster. So let's look at this. That one's growing slow, that one's growing fast. So we can figure out potentially what are the important molecules that determine growth. The mussel, we don't have that asymmetry, so we weren't sure whether we would see this difference. But moreover, we've got this problem. Genetic background is different. They have a different genome. So can you really look at the two? Is this like trying to look at apples and pears? Can we compare them? Moreover, the shell structure is different. So can we really use this model? So we started to look a little bit more at the characteristics in terms of the genomes, the chromosomes, and they're quite disparate. This is the mussel, there's the oyster. There's not a lot of match-up between them. A much bigger genome, many more genes in the mussel, and so on. We look at the proteins, that's the business end. They're the proteins, they're the pumps, they're the channels, they're the receptors, they're the hormones. We look at the proteins in general, there's only about 40% conservation. But when we look at the specific proteins to do with building shell, here we have 80%, 70, 80% conservation. So we have modules in the proteins that are highly conserved. So this seems to be quite a conserved system. Moreover, if we dig into the more lower structural level in terms of the shell, then the general organization of the framework and the calcium carbonate is quite conserved. So having looked at this overall picture, we said to ourselves, okay, maybe we can work with this, maybe we can find some commonality, and maybe we can figure out what might be determining growth of the shell under a normal environmental situation. So we went and we said, okay, so we'll look across the gradient. So here's the great advantage of these massive sequencing that's ongoing. There are many, many genomes now available within the databases, many transcriptomes. So this allows us then to actually go a gradient, to go from perfectly symmetrical through to increasingly asymmetrical, to use all that data, to filter all that data, and then to try and come into some very common conserved molecules. And so this experimental workflow, I'm not even going to try and detail here. It's just to give you an idea of really what we wanted to do. So the first of all is just to make this comparison. How are they similar and dissimilar at the level of the genome and transcriptomes of a perfectly symmetrical and asymmetrical? Try and pinpoint those genes and those non-coding factors that may be important, that are popping up as being common in different organisms that we have here. But having that doesn't really give us an answer in terms of asymmetry formation. It just gives us an association. So then what we want to do are the functional studies where we can have a causal relationship between the gene by eliminating it and the growth. Can we show that? So I'm not going again into detail. All I want to do is highlight this point here, that when we did all this filtering of these different genes, we came up with a series of candidates, okay? Not a very long list, which is good, because if we have 2,000, we can't handle them in the lab, much fewer. Our filtering method picked out the known shell-related proteins. Therefore, that suggested to us that the filtering approach we took was fairly appropriate, and that we were managing to filter the core. We found a lot of unknowns, and the biggest surprise for us was that we found a lot of non-coding genes in our differential set that seemed to be linked to this asymmetrical growth trait. So these were the 2 points. 1, we identify the candidates, and 2, suddenly we're aware that this new regulatory level that we weren't thinking about, which are these non-coding RNAs. And this, I think, from my perspective, is going to be very, very important in the future. So jumping now to the function, forget all the rest. We did a whole series of validation, but just to jump to the function. And as I said to you, it's one thing to identify a putative gene or a putative regulatory factor. It's another thing to actually demonstrate that it is functional. And so this is what this slide is showing you here. So we just picked out 2 of these long non-coding RNAs, so these regulatory factors. We had mapped onto the genome to understand what they might be regulating, which genes. And then we named them. So this one is regulating TIMP, which is tissue inhibitor metalloproteinase. This is to do with turnover of tissue. And SMP, so this is a shell matrix protein domain. And we said, okay, so here, first of all, to talk you through it, this here are the controls, okay? This is the round shell, this is the flat shell, okay? So if we were to wait for these to grow and try and measure them with calipers, we'd be there for a year doing that. And our method for knocking out genes would not last. It only lasts for about a week. So we needed to devise an experimental setup that would allow us to study growth within a time frame which is reasonable and that is easy to observe. And therefore we came up with this model, which is the drilling model. We drill a hole in the edge of the shell and the animal will repair it. And as you can see in the round shell, which is here, this is the control, and this is one where we give it this random mix of nucleic acids just to simulate our knockout. And as you can see, the round one grows perfectly. It grows across, in the case of the mussel, in the case of the oyster, it's growing across about 5 days. But on the flat one, look, the holes are completely empty. It wasn't able to repair the shell, okay? Not in that time frame. It would need much, much longer to repair those holes. And then we said, right, so now let's try and eliminate those long non-coding RNAs that are regulating those genes to do with the shell. So that's the remodeling, the metalloproteinase, and the shell matrix protein. If we can knock out the long non-coding RNA, then we stop the inhibitory effect on those 2 genes there. And so this is what these 2 panels represent. So again, we have the round, and then we injected into the flat side these specific molecules to inhibit the availability of the long non-coding RNA. So it couldn't go and lock onto the genome on its target. And therefore, it would stop the gene production and stop the protein production. So again, in the same time frame, you can see when we do this knockout, this is represented. The round ones are growing and filling their hole. Okay, that's why they're slightly white. But when we look in the flat shell, you can also see that this has actually grown. So we showed from this that in fact these long non-coding RNA and these 2 particular proteins, that are encoded in the genes that the long non-coding RNA is binding to, are fundamentally important for rate of growth. So this suggests to me that all of the previous list that I showed you here, now we need to go and screen to try and dig in a bit more to what they're actually doing. But this is now our experimental paradigm, because this is the way we can actually test that. And then we just said, okay, let's just go check what the actual shell looks like and how these particular genes would normally affect the growth of the shell. And that's just to give you a feeling for that. This is what it would look like when we look at the crystals close up to the repaired area. It's less clear. I don't see it so well from here. But basically what happens is when we block these genes, the crystal shapes change. Okay, but this is not a straightforward story because it doesn't change identically in the two sides. So we know that these long non-coding RNAs act on specific genes that are linked to turnover and linked to the shell production, and that when we remove inhibition, then that stops inhibition of growth. So the story is much more complicated than we initially thought, and we know that it's modifying the actual production of the shell. So now, of course, we need to do a lot more work, as is always the case, to look in much more detail at this regulatory circuit. So to summarize, in fact, the shell growth is regulated by a very small number of genes. There were only 109 that we found that were differentially expressed. Some of them are well-known shell matrix proteins. So that was good. So we're seeing what we would expect to see. But we also found new proteins, and we also find these long non-coding RNAs, which was not something we were expecting. And that in the asymmetric bivalve, in fact, it's the long non-coding RNA, which seems to function as this cis regulatory module. So it's coming in and acting on another gene, and that this then shifts the balance in how the genes are expressed and how that framework for the shell forms and how, therefore, the shell is created. So that's the kind of first chapter. But the question remains, how resistant are shells to acidification? So this is the background information to be able to understand. Of course, now we need to look at how acidification is affecting this system. And we haven't actually done that yet. I need to get some more funding. But clearly, this system is very, very appropriate for asking at a level of the organism what's actually happening. And this is then where it leads into this second part here, because actually, as I said, the majority of studies either look at the effect of OA in relation to shell dissolution, so how does it dissolve, or how does it not form in a larvae. But there are very few of them that question maybe the physiological consequences and therefore how changing the physiology will affect the shell growth. And maybe it's more important, the physiology, than the actual shell itself. So this was the second question that we had. How do they actually respond to change? We know that the shell growth changes when they change food. So if they have less food, they grow less. Or if there's a change in the environment, they'll grow less as well. So, but how does that happen? How is the signal from the environment or from within the organism transmitted to the shell? And how do you keep the two shells in a bilaterally symmetrical animal growing at the same rate? Now, this is my other hero here. This is Claude Bernard. And he understood all this time when he started this area of physiology that to survive, you need to have a fairly constant internal environment. And that concept is a homeostasis. It's whereby you have a corrective mechanism. We're familiar with this when you talk about a thermostat in a room. You set the temperature. If it goes slightly above the temperature, it switches off, comes down again, and trips the switch when it gets too warm or too cold and comes on again. And the organism is very similar. It has endocrine factors or nervous system that can be triggering this cyclical. So we have what we'd call the sort of the basal, the normal range, and then the way it works is through this oscillation. So we questioned it, when we look at the bivalves, it's such a simple organism. Do you really think it has a nervous system? I mean, it just looks like a blob. But in reality, they're very complex organisms. So we asked ourselves, is it local or central? Of course, there needs to be central, because to synchronize two shells to grow at the same rate, there needs to be central. But we did ask ourselves, local, because we know at the mantle, level of the mantle, this is regulating the growth. So this is some very basic concepts, autocrine, so self-regulating cells, or paracrine, little groups of cells that are regulating each other. And this would be occurring here in the mantle. But then we actually look at the nervous system. It's actually rather complex. You've got a nerve net and then ganglia. So these are like the precursor to our brain, but instead of being all clustered up on the top, the ganglia tend to be sitting around the body. And our particular model is the most simple, which is the bivalve. As you can see, there's much less of this nerve net, and primarily we have this main circuit, and these little black blobs represent the ganglia. So this is like the equivalent of the brain. So this is what we then tackled next. Is there a nervous innervation, and is there a nervous regulation of that process? Because we need to then understand that to then look at how ocean acidification or change in temperature might influence the regulation of the mantle and therefore the production of the shell. So the first thing was to try and map that. And so what we did very simply, and just zooming in a little bit so you get a better idea about really where the growth occurs of the shell. So this is the outer mantle edge, this is the middle mantle fold, and this is the inner fold. So this is the bit, the outer edge is the bit that we're really important for generating the shell, because this is right next to the extrapallial space. So we wanted to know how innervated that is. And so we did this immunocytochemistry. As I said, there's no reagents, so you have to really either make them yourself, or if you're lucky, something like 5-hydroxytryptamine. So it's a very conserved molecule, and therefore your antibodies that work in mammals work also in a bivalve because the actual molecule is identical. So we try to map the nervous system under different kinds of conditions. And this is just to demonstrate to you. You look here at the green, which represents the 5-HT, you have this massive green coloration. So this is highly innervated, this outer mantle edge. So this is the edge that's important for the actual growing of the shell, is this bit here. And this is the bit, as you can see, very innervated. This less, inner less, okay? Most of the action is happening in this, in that region there. So having seen that, we then went on to the next experiment, asking about the local and the central regulation. So again, we came back to this model, which is the drilling hole model, because this will grow across like 20 days. So you can actually monitor the growth process. Your peptides might be stable if you've amidated them over a week or so. So if you inject twice, you can expose the animal for sufficient duration. So we set up again here the model. Here I'm actually looking at the muscle. And so we drilled in the right and the left. And this is just to show you what happens. In blue and pink, to the right and left. And as you can see, this is the regrowing shell. So by about 10 days, 60% in the muscles has regrown. And as you can see, the rate of growth in the right and left is the same. It's at the same speed and in the same manner. So then we said, if the nervous system is important there, what will happen if we try and break or interrupt the nervous system? So we severed this cerebral ganglia, which is the ganglia that's more important for innervating the mantle. And severed that, we severed the connection between the two sides. By severing that connection, you can see we stopped the regrowth completely. So it was unable to regrow its shell, it just stopped. And then we went to the mantle, and we're not looking in the mantle at the nervous system, we're looking at these mantle cells. There will be nervous tissue, but we can't really separate the two things. And this is PCR, looking at some specific factors that we already know might be involved in calcium regulation that are present in the genome and present, therefore, in the bivalve itself. This represents the time zero, and it's all the same color. So that says everything is at the same concentration. So we've got calcitonin, important calcium regulatory hormone given to women postmenopausal to try and control the bone density. We've got CCK, which is not really involved in this. It was like a negative control there. It shouldn't do anything. And then a series of other peptides we had identified in the mantle. This is the case where we haven't cut the nervous system. And as you can see, the drilling and going and looking 10 days after, we've got a massive shift in the expression of these proteins here. So this is what it looked like before. You see they're all a very similar color. And when we get this color gradient, the red means highly, highly modified, highly expressed. So these group here of peptides really changed. When we sever the connection between the right and left, therefore the cerebropleural left and right hemisphere, we completely change that. And in fact, those that were important before basically disappear. Okay, so what does this tell us? This tells us that the nervous system innervates the mantle, and it tells us that the intact nervous system is required for the regrowth of the shell when it's damaged. And it tells us there are local peptides, which we're not yet calling neuropeptides, because of course they may be produced locally and the nervous system may be triggering their release. So we know that we have these peptides that are effectively acting on or within the mantle to modify the process of shell production in the extrapallial space. Again, confirmation. Here's an in vitro experiment with that molecule calcitonin. We isolate the mantle cells and we then add in peptide, or we don't add in peptide. In this particular experiment, we've included calcium. So calcium binds to calcium. That means that if there's an increased movement of calcium into the mantle cells, so this was a culture, then it should be greener. If it has no effect, there'll be no change in this green fluorescence. The blue is DAPI, just to look at the nuclei, so we can co-localize and check it's not an unspecific random spot that's mucky. And as you can see here, the calcine in the control is rather muted, but the calcine in the stimulated mantle cells is rather fluorescent. So basically, this is telling us that if we administer these peptides, it actually affects the movement of calcium in the mantle cells in vitro. Okay, it's not in vivo, this is in vitro. And when we look at key molecules, these are enzymes that are involved in that. Carbonic anhydrase is important for creating calcium carbonate, doesn't change, okay, but alkaline phosphatase does, and that's involved in shell remodeling. So somehow or other, the calcitonin is triggering a modification in calcium uptake, but it's also triggering a modification in some of the enzymes that are to do with shell building. But this is in vitro, so it's not actually really a whole animal. This is just part of it, and of course, you can't really say that that represents what happens in reality in an intact organism. So then we went to the intact organism and we said, so now what we'll do is we'll cut the connections so they cannot regrow, but then we'll introduce the peptide to see if they can repair the shell in the whole organism. And so this is what I'm basically showing here. I'm not showing the experiment per se. What I'm showing you is the results. This is scanning electron microscopy. And what we've tried to do here is to actually look at the hole that we drilled. And then we look both at a higher magnification at the crystals. And we also use FTIR to look at the presence or not of calcium in that regrown shell. What do we see? So control, we didn't do anything. It grows normally. When we give PPS, it doesn't stimulate growth because we've cut that ganglia connection, so it's not able to regrow. But when we give the calcitonin, we've got different forms, then they really do regrow, okay? And this is our negative control, because this is just a peptide that's not associated really with this process. And it creates a protein mass, but it doesn't create a calcified crystal structure. It's just proteinaceous. So I think you can agree this is what the control looks like. And when we give the calcitonin to an animal that can't regrow its shell because we've cut the ganglia, it's able to regrow the shell. Of course, there's a lot more to it than that. So we continue. It's just to let you know, again, long non-coding RNA is involved in this complex process. What I showed you is just one very small part of the whole story, and there's a lot more work to do here. So I'm going to speed up because I suspect I'm overgoing my time, and I will quickly finish. I've got another couple of slides.
Don't worry.
So in my summary, so we know that shell growth is controlled locally and centrally. We know that, therefore, the central system, these lobes that they have, these ganglia, surely pick up and then translate into a signal through the CNS to the mantle, information that then modifies the cell growth, and that really in the mollusks, the long encoding RNA are very abundant and seem to be very important regulatory layer. And then I come to this question, which is the Anthropocene. But what are the regulatory boundaries? So this is what I'm saying, is that you need to understand the animal physiology if you want to understand how it will or won't resist changes in the environment. And the shell may dissolve, but that's going to take a lot longer than an animal dying if it cannot resist the change in the environment. So I think that the key focus should really be understanding the regulation of the animal, because it can die in 12 hours, whereas the shell will take years to dissolve. And so this very quickly, I'm not going to go, this is just the whole story on the carbonate. It's just basically saying that as we get more saturation, it becomes more difficult for these calcifiers to calcify. What this tells us is that actually, and this was an experiment under OA where we put in mussels, oysters, we looked at regrowth of sanded shells, and we looked at growth. And basically what it tells us is that under OA, they stop growing. And that's true for the oyster, and that's true for the mussel. So when you put them under OA conditions, they stop growing, but they keep feeding at the same rate. So the energy demand is much higher. They need to expend more energy to maintain themselves. That may be maintain shell, but it also may be to maintain basic physiological functions. When we look at the molecular side of things, and I won't go in detail, I think what I just want to draw attention to here, this is all, looking at gene expression, but look how different our PCA is, or PCOA is, when we look at the Gala provincialis, the mussel, a highly invasive species, very successful, invading everywhere in the ocean that it can, and the M. gigas, which basically you don't really see much difference between controls, ocean acidification, or anything. Gala provincialis, you see a big difference between OA, and there's two groups there, one with the hole in the shell and one with just exposure to ocean acidification. And you see a very different response from the control to the exposed, okay? So there again, I'm saying these bivalves are not going to behave in the same way to OA. And that's just two. And there are 11,000 of them. So I think it's very difficult to predict what are going to be the consequences of ocean acidification. And sort of the final point here is when we look at the gigas, in fact, in reality, it has a very low response. This is 60 days. Most experiments look at 24 hours, 48 hours, maybe a week. This is 60-day exposure to OA. The Mytilus still has a lot of changes in its expression levels. It seems to have a new basal level of expression of genes. I don't know if that's good or bad, because they both don't grow. Maybe the oyster is more adapted to these more acid conditions, or maybe the Mytilus is more adapted. I don't know. I can't even tell you, because we haven't really taken this further, but we need to if we want to understand the benefits or the capacity to resist between these two species. So to conclude, we have this very variable shell construction, and we have a massive physiological plasticity across the bivalves, because they're very long-lived and their genomes have undergone a very significant modification. I think that this means that ocean acidification is going to have very species-specific effects, and you can't make a single rule and say it will apply to all of them. We have a very high recruitment of genes to a new steady state in Mytilus gala provincialis, so the mussel, compared to the oyster. But I don't know if that's good or bad, because we haven't done further experiments. And the physiological limit, how low the pH can go. What we did was put the pH to what would be expected in 100 years' time, but probably now is 80, 60-year time, because the speed of change is accelerating. I don't know where the limit would be, if there is a limit, since, as you can see, if they came in the Cambrian and they went across all those high CO2 and very modified big ranges of temperature, on a geological timescale, maybe the genome has the capacity still to allow them to respond to this much, much more rapid change that we're seeing now. So on that point, you see I have more doubts than I have answers. I'm very sorry. I thank everyone and acknowledge, of course, this is work done with many colleagues and also funding from a diversity of sources. And thank you very much for listening to me.
Thank you. Thank you very much, Deborah, for this very nice talk. We have time for questions or comments now. So, any question or comment? Not, I have one comment. Well, I noticed that you use the name of the Academy and our symbol in your slides. This means that you prepared your talk specifically for this session. You didn't recycle old talks too.
No.
And this is, I appreciate this. I am an entomologist, and as you know, your subject reminds me the formation of the elytra in beetles. Elytra are hardened wings, and in the elytra there is a combination of mineralization and organic compounds. The proportion is different, than in mollusks. It's about 60% of minerals and 40% of organic. But this is quite well studied, and perhaps you could perhaps be inspired on how. Of course, it's very different because an elytron is simply a modified wing. The precursor was the wing. I am wondering about your two models, mussel and oyster, the basic principles of regulation, matrix proteins and non-coding RNAs are similar in both models. There are general rules.
So I didn't show that. So that's a very important question. I mean, I have to say, the work that you did on the small miRNA, you're interfering with mini RNAs. In some ways, this was what we were looking at initially, remembering the non-coding, because this was, in a way, serendipitous. What happened was, we had done some sequencing, which was very poor. So I said, no, we can't advance with this sequencing. We need to resequence. So, of course, then the budget was tight. So then we said, okay, we look and find a good, cheap sequencing. So we did, and we sent it to be sequenced, and it came back. And it was a bit different from what we expected, until we suddenly remembered, instead of using mRNA, so capture of mRNA for making the library for sequencing, we use ribosomal depletion. So in essence, we removed ribosomes, but it meant that everything there that was RNA was there. And this is where we captured all the long non-coding and all the encoding. So in reality, the long non-coding is highly, highly abundant across the bivalve. Very abundant compared with the mammals.
And they are very diverse.
And they are very diverse. So across the ostridae, you get a good level of conservation. And you can manage to map. So we can map common long non-coding against potential targets. But when we go into the other families, it's another whole suite of long non-coding and a different combination. So the conservation of long non-coding is not that good. And then, of course, you need to take the long non-coding and look for the target. So, and this then becomes much more difficult, and it doesn't seem to be very well conserved when we look across all the different bivalves that have a decent genome available. So it looks as if there's been quite a bit of diversification at the level of the regulatory molecules that are these long non-coding. And, you know, I think it's like 30%, 20-30% of the genome that we were seeing seemed to have this long non-coding. So it's very, very abundant.
Thank you. Geoffrey, please.
My knowledge of Bussos is mostly
Eating.
as food.
Exactly.
And also, but also that they concentrate in some areas, they concentrate pathogenic viruses. But this is not my question. My question is that I imagine that the surface of the mantle is a very good site for bacteria to live. Is something known about the microbiota of the surface of the mantle?
That's a very good question. It's always this thing is, what do you look at? We have started to look at that. So we have looked at the mantle, we've looked at the hepatopancreas, we've looked at the gills. Obviously, most of the tissue is being exposed to seawater, but these tissues, in terms of composition, secretion, mucus, are a bit different. And there is a differentiation of the microbiota in the mantle, in the gill, in the hepatopancreas. What they're doing, here I cannot say. Possibly they do contribute in some way to this process, but I'm not able, because we haven't looked at that. It's another whole area that needs to be considered for sure.
Because sure that many bacteria would like to live in an environment in which there is
Exactly.
these different forms of carbon and calcium.
Yeah. I'll be in touch. We could send you the data. You can have a look at the data, see something interesting. I mean, part of the problem is the volumes of data, but surely there is clearly the bivalve is able to modulate the microbiota, because we've been trying to see about this question of immunity and innate immunity, to see whether they do have memory, or if they have no memory and you really need to have an acquired immune system. But certainly when you cause this shift by exposing them to a pathogen, then they will try and reestablish, or temperature, they will, so for sure there is this host microbiota interaction, and it's quite abundant in the mantle. Yeah.
Okay.
Thank you.
Thanks for the talk. I have a couple of questions. One, what about microRNAs in the bivalves? And the other one, I think that you mentioned that they originate in the Cambrian explosion, during the Cambrian explosion, right? So my second question is, what about the CO2 levels? You mentioned in one of the graphics, much higher levels of CO2 before, but I don't remember whether this was before the Cambrian or after the Cambrian.
So first one, microRNAs. We haven't really looked at them. The really compelling observation is the long non coding. And we did debate looking at the microRNAs, making libraries, but we haven't gone there because we were decided the long non coding seemed to be so important that was the most obvious choice in terms of target. But certainly, if we had the money, we would also try and look at that. So this black line here is the CO2. Unfortunately, there's no scale on this. It's one of these, as you can see, it's not the most beautiful picture. I am not one of the students are amazing because they can make these amazing pictures. I'm rather rudimentary. I like just to have a clear picture and show it. So this is what you see is happening to the carbon dioxide across time. And this blue line is what's happening to temperature. So actually, they really emerged when there was this really massively high level of CO2. And they have persisted through high and through low. I'm guessing here maybe there was a big die-off. I was trying to look a bit at the paleontology to understand where the fossils are and where they're changing. But, current day levels are very low. They're at this level here.
Do you know if there is a correlation in the fossil record between the levels of CO2 and the shell thickness, for example?
No. There are studies on looking at what particular, families or taxa are coming at different time points, ones that appear, ones that are lost, ones that are long-lived, ones that are short-lived. But I haven't seen really measuring, because sometimes they're kind of completely like a pebble inside a piece of stone. So I haven't seen that. There have been studies on modern day ones, looking at the thickness. Not very convincing, where people leave them for much longer. Maybe if you want to dissolve it, just put a bit of hydrochloric acid. But I mean, hydrochloric acid is nowhere near seawater. It's very weak acid. Carbonic acid, very weak acid. So the studies are not compelling of current day bivalves in terms of putting them into pH 6 water.
Because the bivalves are very easy actually to maintain in the lab.
Yeah.
So it would be easy to, probably some people doing that, to look at windows of sensitivity during early development, whether exposing them at different levels of CO2.
I think there's two things here. It's easy but not, because if you're in the ocean acidification, they don't accept anything unless you have 3 independent circuits. So you need to have 3 completely independent circuits. Yep. Otherwise you don't publish. So anything, all work to do with ocean acidification is really, you have to have independent circuits, 3. You have to have titration of carbonate, alkalinity daily. So it's a massive job to do this, because so modifying pH, then of course your CO2 coming off the water, so you have to then put a lid on top to try and stop that, otherwise everyone's complaining about you gassing them, and the cost of all this CO2 coming off out the bottle. It's actually technically not very easy to do that, tricky. And you know, you see these guys with the aquaria for the corals, they have an enormous difficulty maintaining their corals because they have to get the right conditions there. So this is a technical issue. And the larvae, I can say, and of course this is my other passion, is the metamorphosis. So the larvae are very interesting. They're very tiny, they're pelagic, and so they're floating. And then they are feeling for substrate that's good for them to settle on. They have a foot that's probing. They're not at all calcified, and they are micrometers, they're tiny. Once they find a surface, and they feel it and they find it a good surface, and it needs to have, in fact, it needs to have a biofilm of particular bacteria for them to then settle and fix. Then they will start to form, so they'll lose the venom, this floating structure to allow them to go through the water. They lose the venom, and they start to form the shell. And they have the shell secreting gland will then start to secrete that. This is probably the major problem, because the saturation is lower, is higher down there. So it's less available, calcium, and they need to be able to produce it de novo, this shell. So they need to produce the protein, and they need to produce the calcium carbonate. And so I think they are very vulnerable. But there are a few studies, but very few about how vulnerable they are.
One last question. Some years ago, there was debate on the presence of aromatase. Now I'm going back to endocrinology.
It's all rubbish.
It's not true, right?
It's absolute rubbish. It's not true.
It was a false aromatase activity.
Totally.
Right?
No, I mean, there's a group of people still. I mean, Sandy Scott wrote a review in his retirement, in his 70s, showing that there is no invertebrate, at least these bivalves, with aromatase.
But still, the tritiated water assay still was giving false results.
Well, the problem is the following. If you put your hands in water, if you're a female, you leave a lot of estrogen in the water. So that's the first point. Post-menopausal women, where is the main source of estrogen? The skin. Do they have the signaling? Do they have the receptors, the nuclear receptors? Yes. Maybe not estrogen. Well, in fact, they do have estrogen. Can they take up estrogen from the environment? Yes. Can they produce it? No. So, I mean, of course, you might say that the system is built to be like that, because it's the external signals in these organisms that they take up that then trigger whatever they do. But they do not produce it. But it doesn't mean they don't use it if it's available in the environment.
Thanks.
Any other comment or question? If not, I will. Before closing, I will give you silver of the Academy.
Oh, thank you very much.
You
Oh,
should wear it proudly
I will wear it very proudly.
in all your academic
Exactly.
activities,
Yes.
also social activities, familiar activities. We are very proud of you. And
Thank
should
you
be
very much. I'm very proud.
of
Thank you.
the Academy.
And I need is the address so that we can put it on our, you need to give us the card so we put it on our publications when we, our affiliation.
Okay.
Thank you very much. Thank you.
It's in silver.
It's wonderful. Thank you very much. Now I have to be very careful not to lose it.
After that, we can close the session. Thank you again for coming.